and I will be sharing the... Okay. So we'll start with the first question. Do you see my screen? At least please one of you answer so that we are on the same page. Okay. Okay, great. So as for now, do you understand? Yes, no. Oh, there's something missing here. Not why. I don't know why this is happening. You guys see what is what yes and what is no? For me, it is not displaying. You can't? Do you see if this is yes and this is no? Apparently it should be. Well, let's take one as, well, I can't, it's too late. Oh, terrible. Let's assume Maggie is assuming that the top is yes and the bottom is no. This is terrible. Why it's not showing the... I hope it's not the same for the other questions. So one is yes, two is no. Please change your answers if you displayed already. One is yes and two is no. So 50% of the students don't know yet what are the basic steps of the qPCR. Okay, we have a problem here. We have a problem because we spent 2 whole lectures describing this with the recordings. For those who are not able to understand the qPCR, please, why you are not showing up in the office hours? Why you are not staying after the class to ask those questions? So I can help you as much as I can if I know that there is a problem. If I don't know the problem, then there is an issue. Okay, so let's move to the next question then. If you were to choose between Cyber and Tachman probes for a qPCR, which one would you choose? You are only 23 in the class today, and I would really appreciate if you answer, even if you don't know 100% the answer of the question. Okay. So Cyber and Tachman are both good. You have to evaluate if they are expensive. You go to Tachman, and you have to also evaluate that Tachman is more specific. So Tachman is much better to go if you have the money. Between Cyber and Tachman probes for qPCR, which one is more specific? I already answered. Recall that you have that probe in the TACMAN that gives you specificity toward that sequence within the gene that is not related to the primer sequence only. On the other side, cyber, you rely on the specificity that comes from the primers only. So if the primers bind somewhere similar to that gene, is not going to be as specific. That's why TACMAN is more expensive, more specific. Less expensive, less specific. Okay. What is the primary goal for creating a knockout animal? So when you're reading a question, please be careful between the details. What is the primary role or goal of creating a knockout animal? Increase the expression, introduce foreign DNA to the genome, eliminate the expression of a gene, and enhance tissue-specific gene expression. Pretty good. Now, this question can be problematic if little bit more details are added. And let's see the next one. What is the key advantage of conditional knockout compared to a standard knockout? Okay, so is this to increase the expression of a specific gene? Is it to allow the gene to be inactivated in a specific tissue and time? Is it permanently to inactivate the gene in the embryonic stem cells? Or to prevent off-target effects of a CRISPR technology? The answer is it allows gene to be inactivated in a specific tissue at a specific time. So the conditional knockouts offer you this practicality that the general knockouts do not. Next. When generating a cDNA library, the cDNA molecules are single-stranded, double-stranded, hybrid RNA DNA molecules, or all of the above. So recall the cDNA library, where it comes from. And what is it? This is one of the confusing questions that you can get in the exam because it's not super clear what exactly I want. And in this situation, I would really like that you guys comment back or like write note, oh, this question is not formulated correctly. Because that helps me to be more careful when I pose such questions. Okay. What do you think about the answer? Is it a hybrid or it's a single strand or double? This is a mistake. Sorry. This is a double stranded. It's not a hybrid. So again, the way I put the question, I know it's intentionally made to be confusing. When generating a cDNA library, the cDNA molecules are, it could be single-stranded because you are making one copy. It could be double-stranded because you are making the complementary part of that RNA. And at certain point during the first step of the cDNA library, you are really converting all the RNA into DNA. And now you have a molecule which is RNA DNA hybrid. The question should have been formulated better, saying what is the end result of the cDNA? The end result should be double-stranded because you need to clone that into a vector, remember? And you can't clone single-stranded DNA into a vector. And you can't clone a hybrid RNA DNA molecule. So the right answer should be this if the question was better. Okay, let's go to the next. And what is the reason behind using a tissue-specific knockout rather than a conventional knockout? Again, when you're reading a question, and for those who are going also to the medical school, you will encounter some questions like, you have to really be careful when you read the question. The question is related to using a tissue-specific knockout. So there is part of the answer already in the question. Is it to study the gene function in a bacterial system? Yeah, probably, but why in the bacterial system? To reduce the time required for genetic manipulation, is it true? Because we know that we have to take more time to generate a tissue-specific knockout compared to a conventional one. And is it to avoid early lethality caused by the absence of the gene in all tissues? This sounds more logical. And the last one would be to increase the expression. Increase the expression, be careful. It's not to increase the expression. So for those who got confused with this guy, this could be a part of the application. Why would I generate a knockout animal to go test this in a bacterial system? It doesn't make sense practically. It's confusing that yes, you can do that if you want, but instead of making a transgenic animal, just take the plasmid and the vector and go to that bacterial system and express it there. Okay, makes sense now. And if you would like to choose between two different techniques to study the RNA, would you choose an RNA or a microarray? I would choose microarray based on certain criteria and RNA-seq for other criteria. So this could be also in a format of true or false. Okay. Such questions in 45 seconds to 1 minute, probably you should be able to answer. That's how I calculate the time for how many questions would be in the final. 17 out of 25, please. Go ahead and answer, even if you are not sure. This will at least give you some feedback. Where is your mistake? Okay, and the right answer is still the majority answered correctly. So microarray is a cost-effective, capable of detecting all genes, including the novel one. This is not correct, because in the microarray, you have to know already the probes you are hybridizing to that microarray. And if we don't know the novel genes, how would we be able to detect them? We need already to have a probe that is hybridized on that microarray. So novel genes are not detected there. RNA-seq uses pre-designed probes and can only detect previously identified gene. That is what microarray can do. So these just are flip-flop between microarray and RNA-seq. Now, microarray, it allows for high throughput analysis and is suitable for identifying unknown genes. Yes, here probably there is a small part where you can consider it's a high throughput analysis, but it's a very minor scale, but it's definitely not suitable for identifying unknown genes. We should know the genes and their probe. It's like kind of similar to number one, but it's formulated in different language. RNA-seq can provide unbiased approach because you can take all the RNA from including microRNA, long, any type of RNA, and you can detect any genes, including the novel ones and the already known ones. Okay, so that's the powerful usage of RNA-seq compared to microarray, because remember, in microarray, you are limited to the number of the genes that you can study. Okay, so we will continue doing this. I think this is the last question. Yes, this is the last question for today. So we will move today to discuss the first application of the protein analysis. Could you please confirm that you can see my PowerPoint? Okay. No, you can't. Okay, you can't. Stop. I'm stopping this and I'm sharing. How about now? Good. Okay. So please, if I'm going, and I noticed in previous lectures when I reviewed the videos later on, sometimes I switch between windows and I start talking about something and you guys didn't see the second screen. It looked so weird what I'm saying, because definitely you didn't see the right screen. So please, if you see that there is a screen that I'm trying to explain and it doesn't show up, stop me. Don't hesitate to stop me. And don't worry if you will speak in the recording, because then I will clip it out. I will not keep your voice there. Okay. So in the previous lecture, we described some basics about the B cells generating antibodies toward pathogens. And this eventually revolutionize how we study proteins and how we analyze the proteomes in a particular setup. Because what we do nowadays with recombinant DNA and RNA and protein technology is to mimic what this cell do in the immune system in order to identify a host or a pathogen or let's say a bacteria or a virus. And we do this setup in vitro, so we can identify proteins, we can see the quantity of these proteins in the blood, in fluids, in cells, in healthy conditions and in disease conditions. So today's discussion would be mostly about the antibody generation. And we will start delving into the applications. We will start with the application that doesn't need proteins to be sorted based on their molecular weight. We will just look at proteins that are floating in serum or blood or fluids. So today is the first application, and I hope you had the time to go through this slide. Again, this is pathophysiology from last year, lectures. Obviously, this is very simplified. I assume that the pathophysiology had covered this very well in the immune responses because it is one of the basic steps that our body adapt toward pathogens and infections and foreign organisms or foreign material in general by generating antibodies against those pathogens. Down the steps, I'm not going to ask you what makes a cytotoxic T cell or memory T cell. Important for me that B cell is the cell that generates the antibodies. And for those who didn't have pathophysiology or didn't study this, it's important for you to really go very carefully on these videos because it kind of give you a very good background how the immune system works, how the complement system with the antibodies and immune cells interact. And it give you an indication when there is something that goes off from this immune system, we get the hypersensitivity reactions, including the one that we presented last time, like the blood transfusion, right? And I assume that now you know very well what is an antigen and what is an antibody. And if you forgot, the antigen is a protein, a pollen, a pathogen, a virus, a bacteria, part of that that is in principle detected by an antibody. Now the site or the sequence, the specific sequence that is recognized by that antibody is called an epitope. And obviously one antigen might have multiple epitopes, as you can see here. So obviously this is how we learn these things about antibody generation and how it reacts to an antigen and how many antibodies bind to an antigen. And we came into the types of the antibodies that we have, what are they, which one reacts to which pathogen. And obviously, we discussed that there are multiple types. Well, there are those, again, in the pathophysiology, you should study which antibody come first, which one is more potent or more efficient in sequestering a pathogen. This is not the discussion for us here. The idea is that we use the antibodies in techniques where we can study proteins, okay? So one of the examples I gave you is we could neutralize viruses by antibodies. And that's exactly how it works. The virus comes first time to our body. Our B cells, after many steps of education, they will generate an antibody toward that particular virus very specifically. And if the virus infects us again, then we have antibodies floating in our blood in the best scenario, or probably if we don't have not that much number of that antibody, probably we have memory cells that can quickly generate these antibodies to sequester the viruses. And that's why it takes shorter time after the second infection compared to the first time exposure. I gave the example of the blood coagulation and many others that we really discussed in the previous session. So this is again the blood transfusion. So we have to be careful how we choose the donors based on the antigens on the surface of their blood cells. Pretty much this you studied many times, but this is an example of hypersensitivity. In principle, if you by mistake transfuse blood that is of the wrong type to a personal, that's the type of the reaction you would have. Probably it's called type 2 hypersensitivity. So the idea is when we look at proteins and the way we study proteins in vitro on the benches in our labs, or when we perform tests for particular viral infection or checking if there is some bacteria or pathogens, the idea is that if this is the protein, which looks very complicated here, this is a very long protein, it's like folded, it's long, and you can see here, like it's kind of showing the primary sequence, which is just the amino acid sequence of that protein. The epitope could be somewhere 5 to 15 amino acid or even more that is specifically bind by a specific antibody. So how do we achieve to get that antibody to bind to this specific protein? The first thing you have to keep in mind is if I have this protein as a primary sequence, primary structure, if this antibody detects this epitope and provided that the protein is not degraded into smaller parts, provided that it's still intact as primary structure, you would detect the whole protein with this antibody. So if this is 5 kilodalton, which is or 10 kilodalton or 20 kilodaltons, which is the measure of molecular weight of a protein, this antibody will detect the whole 20 kilodaltons. Now, if it happens that you degraded the protein when you are isolating it, probably then you will have this fragment or that fragment. But ideally, what I'm describing for you thinks in the very ideal way that the protein is going to stay intact. And this antibody will recognize this epitope, which is the primary sequence of few amino acids of that protein. And therefore, it will detect all that protein. Okay, so for the rest of the discussion of today, that's the scenario we are discussing here. Now, this is somehow mimicking what I just mentioned in the beginning of the class, that we are trying to do in vitro what normally our immune cells perform against a pathogen. When the B cells will generate an antibody that in principle binds to a specific antigen. And most of the molecular biology that you will see in terms of protein studying, it is based on this concept. Now, there will be modification of what is the start material and what's the end material and stuff like that, and how we achieve to get to this step, but this is the main principle. So also we discussed in the previous class the difference between the monoclonal antibodies and the polyclonal antibodies. And we mentioned that monoclonal antibodies detect only one specific epitope on the antigen, in yellow here. However, in polyclonal antibodies, the antigen has multiple epitopes similar to that antigen though, but it's detected by multiple different antibodies. So you can already tell that the monoclonal antibodies are more specific because you have only one type that you are using, or one clone of the antibody that you are using. On the other side, you have multiple antibodies. And this is not really a trivial thing because we really face difficulty to find a good antibody that binds to a specific epitope. That's one of, I mean, if some of you are already in the labs, many antibodies have a lot of unspecific targets. So that's why monoclonal antibodies are more powerful than polyclonal antibodies. And we will describe how we will use the antibodies later on. I listed them here, so this will be repeated probably a few times. So first, let's go to the easy, cheap way to generate antibodies. Just before going to that, I want to go back a little bit here. Like when I mentioned, if you would like to clone a gene, you first have to get some information about that gene. If you don't know the sequence, you probably need to perform a library, get the DNA sequence, know what is the promoter, what is the exons, what are probably the introns, all that, and then predict what is the coding sequence. Probably you want to do cDNA library or reverse transcription and those steps. So you would need a lot of information before you clone a gene. And you would need a lot of those information if you are probably generating a transgenic animal using that transgene. It's the same thing here. You would also need to know the sequence of your protein. And eventually, if you are working on this protein, it comes from a gene that is transcribed. So you would need to know what are the RNA transcripts, what are the splice variants, what are the possibility of splice variants for that protein. You would need to gather a lot of information before deciding what is the peptide that you would use in order to generate an antibody against. So the epitopes, nowadays we have softwares that we can give an amino acid sequence and say, could you predict what is the best epitope to generate an antibody against this protein? And probably that software is going to tell you, oh, if you take amino acid 5 to amino acid 18, it's kind of unique to this protein. There is not much known about other proteins that have similar sequence and similar structure. And then based on that, you would choose the epitope. So choosing the epitope is a very important and critical step. And I'm not assuming at your level you would do that. Yes, it needs bioinformatics as well, and it needs a lot of literature and knowledge about the gene, the RNA behind that protein, okay? But say we already have that, because in this course, we can't cover all those steps. Say we already know what is the part of the antigen or the epitope that is mostly or the best representative of that protein, that I can inject it to an animal, and that animal will have an immune response and an antibody regeneration to work. So the way that a polyclonal antibody works is that we inject that protein or a part of the protein that we know it is going to be representative for that antigen into a mouse or a rabbit or a pig or goat, depends on the facility. And then that animal is going to generate antibodies against the antigen. So you can see that here there's an antigen. It's represented by this red thing. It's a sequence of protein, in fact, which has 3 different epitopes. And you can see that the B cells generated 2 different antibodies. One is triangular and one is like rectangular. And eventually the B cells, remember in that slide, when they get educated and specialized to generate antibodies, then there will be two populations. One of the populations is called plasma cell that secrete a lot of antibodies, and the other population is going to be memory cells, which carry the antibodies on their surface. Okay. Now, the population of plasma cells that generate all these antibodies, the antibodies are actually circulating in the bloodstream. Eventually, when we get a viral infection, the antibodies are in our bloodstream. And you can simply check these antibodies by just taking the blood and just looking for the antibodies that have been generated. So the same thing happening here. We injected the antigen, and then the antigen was captured by B cells, and B cells generated antibodies against that. And then the plasma cells secreted a lot of these antibodies in the blood of that animal. So all what we do is to collect the blood from that animal and then isolate these antibodies. Now, since we have more than one epitope on that antigen, it's only two presented here, but it could be 5 or 6. What will happen is you will have at least in this representation 2 antibodies that are generated against the same antigen, and therefore 2 different epitopes, and that's how we get polyclonal antibodies. Okay, so the polyclonal antibodies are generated by different B cells, so more than one B cell, and each clone of this B cell is going to be directed toward one of the antigen epitopes. It's cheap and we can get quickly the antibodies, but yet it's not very specific, and that's why we use it in research diagnostics, but we can't use such antibodies in therapy. So if we have, for example, an issue that we would really inject antibodies to a human, and this is now in clinics, we have to make sure that the antibodies are monoclonal, not polyclonal. The monoclonal antibodies on the other side is a little bit more complicated, a little bit longer in the procedure, and more specific. Now, in order to generate the antibodies, these steps are similar, but there are more steps that are included into that. As you can see here, we just injected, we collected and isolated the antibodies. It's very simplified, as you can see here. There is much more steps that are not included in this slide. However, when you injected that antigen and you get the blood, most of the time you have to inject the antigen probably more than one time. So keep in mind that the representation of injection only one time is not enough. The other thing is, every time you need to generate that antibody, the polyclonal antibody, you have to start this process by injecting the animal. Okay. In other words, there is no continuity of the regeneration of the polyclonal antibodies. This is a little bit different in the monoclonal antibodies because we want to have some sort of continuity of generation, and that's where hybridomas come into the scene. So the first step would be, again, choose the right epitope of the antigen or the part of the antigen you want to inject to the mouse. And then you inject to that mouse or rabbit or rat, and then you will have the reaction. The B cells, like in this slide, you have only 2 B cells presented. In this slide, you have 4 different B cells that are generated. So 4 different B cells reacting toward that antigen at different sites or different epitopes. So the first step would be to isolate those B cells from the spleen or the lymph node, because that's where the B cells are residing. And then we will separate them in culture with myelomas, which are B cells. And eventually there will be a fusion process because, you know, normally our B cells should react to myelomas because these are cancer cells and should be eliminated. And in this sense, we will generate a fusion of those cells that has the ability to generate an antibody specific toward the antigen we injected. And at the same time, we will have the ability to grow those cells for a long period of time because the myelomas are immortalized cells. So we acquire the ability to generate antibodies side by side with the abilities of growing for a long, long period of time. And now what we need to do is separate each different cell, B cell, or fusion of B cell that could react toward that antigen. So we have the B cell that has the pink antibody fused with the myeloma. So this is hybridoma number 1. So it's clone number 1. And the blue one is another clone. The green one is another clone. The light green is another clone. And therefore, all we need to do now is to grow these cancer-like cells, my hybridomas, that are generating those antibodies. And if we take, for example, this cell and put it there, and this one, I put it here, 1, 2, 3, 4, each one of them is generating a different antibody toward that antigen. Okay? And that should be very specific to the protein. Not necessarily that all work very efficiently, though. We have to test that. But we are sure that each of these clones here are specific to one epitope of the antigen. So that is the step which makes things a little bit longer. First, fusing the B cells with the myelomas to make the hybridomas. And after that, to isolate each B cell separately and generate antibodies out of that. In this particular slide, they took only the light green one, and they show you that they grew it and they cultured it. And in the culture, they collected as much antibodies of the green as they need. Okay. Now, how they do the screening and all this step, this is not something we want to go forward in the details. It's important for you to know the difference between monoclonal and polyclonal antibodies. And that should be sufficient for this course. So monoclonal antibodies, as a summary, they bind to a single epitope of an antigen. And they are generated by a single B cell, as you can see here. Each one of this is a clone, and it's generating an antibody in green against that antigen that we injected here. Okay. Now they are more expensive, obviously, because you can see we have to have not only injecting the mouse, we have to have all the study to identify the antigenic part, the mouse to inject or whatever animal. Then we need to take cells, B cells to grow. On the other side, we should have cancer cells, the myelomas. Then we will generate the fusion cells. Then there will be separation of these fusion cells into like single wells. So that's extensive work, and that's why it's more expensive and more specific. And these monoclonal antibodies are used in any field, including research and including therapy, so they can be injected to human if needed. Now, let me see. Is this gonna work?
Empower your students to be more authentic, impactful writers with Grammarly's comprehensive.
Okay, so let me quickly share with you my slide to see.
The hybridoma technology is a common method to produce monoclonal antibodies. Monoclonal antibodies, in contrast to polyclonal antibodies, originate from a single B cell and are identical, which means they bind to a single specific epitope on the antigen. In contrast, polyclonal antibodies have been produced by multiple B cells of different origin, and those polyclonal antibodies bind to different epitopes on an antigen. Monoclonal antibodies are often used as therapeutics, for instance, against certain cancer types. How those monoclonal antibodies are generated with the hybridoma technology will be explained in this video. Before that, around 98% of all viewers have not subscribed to the channel, so please do so to support the channel. I greatly appreciate your support. In the first step, the antigen of interest is injected into a mouse, provoking an immune response against it. B cells recognize the foreign protein and start to produce antibodies against the antigen. Plasma cells inside the spleen of the mouse, so-called splenocytes, can be isolated and cultivated in cell culture. Those splenocytes have the ability to produce antibodies, but the problem is that they are not immortal and therefore cannot be cultivated for long. To solve that issue, the plasma cells have to be immortalized. To this end, so-called myeloma cells come in handy. Those cells are immortal cancer cells that can grow indefinitely. Plasma cells and myeloma cells can be fused to generate a hybrid cell type of both, referred to as hybridoma cells, which have both characteristics of interest. Hybridomas produce a specific antibody and are also immortal. Hybridoma cells are cultured using a selective medium that only supports growth of those cells with both characteristics. In the following steps, single cells from the culture are isolated and grown alone to generate clones. Since each well contains antibodies derived from a single hybridoma cell, the antibodies produced are considered monoclonal. The specificity of those antibodies can further be evaluated with an ELISA, or it can be analyzed using Western blot. Here are two videos that briefly describe both of those techniques. Feel free to check them out. Also, don't forget to like.
Okay. So with this, we have a good comparison between how we generate monoclonal versus polyclonal antibodies. And just for your own knowledge, we should know that there are other types of antibodies that are not generated using these two systems. And these would be genetically engineered antibodies, or what we call recombinant antibodies. Now, instead of injecting an animal in order to generate a polyclonal antibody, or generating an antibody after selection of a particular B cells to make a monoclonal antibody, you can simply amplify a DNA that you would know that could be complete, like the DNA for an antibody in a cell that could generate antibodies toward a specific target. So in other words, there's a lot of things you should do in order to reach cloning that particular DNA. I'm assuming this is going to be a bit difficult for you, but it's good to remember that with recombinant DNA technology, one of the applications that you can do is, instead of overexpressing a gene, you can overexpress the DNA that is, in principle, allows you to generate antibodies. So you would overexpress the DNA, or the light chain, the heavy chain of that antibody, and it could be specific to a particular protein of interest or antigen. So this is something we do. There's more reference information for you here if you're interested to study more about how recombinant antibodies are generated, please refer to that page. Now, just in terms of what do you need in order to generate the recombinant antibodies? Of course, again, exactly like I mentioned before, you need to know a lot about the antigen, about the RNA, about the DNA sequence. And say, for example, you want a starting material to generate a recombinant antibody. One of the material that we can use is, in principle, those hybridomas. Like if we get to this stage here, where we have hybridomas in those cultures that are generating antibodies, if I would take the DNA from those cells, then I can predict what is the antibody sequence that they are generating. And therefore, I can get that DNA cloned into a vector, and I can introduce it into cells that can generate antibodies. And in fact, that's exactly what this slide is telling you. You can start from cells that are generating antibodies. You can get positive B cell screening at the single clone identification level. Then you can amplify by PCR the light chain and the heavy chain of a particular antibody. And then you can clone those into plasmids, the light chain and the heavy chain. And if you transfect the mammalian cells with the light chain, heavy chain, those cells will generate the antibody for you. So that's a recombinant DNA technology that you could generate antibodies with. Okay. Nowadays, all these antibodies are available and we use them in research. So in principle, as for now, you can easily understand that when we generate antibodies, they are specific toward an epitope in a protein. So how can I use this? This is the next step in the application process. How do we use antibodies? And how can I detect antibodies? So the first thing you have to remember is that antibodies, they have this specific structure where it binds to the antigen in one side and on the other side is where the complement system normally recognizes the antibodies in an immune response. So in recombinant DNA technology and in novel technologies we have side by side, we are able now to label DNA, RNA, and proteins. So you saw that we could label DNA and sequence it. We could label RNA when we make the microarray. Remember, we had the red and the green fluorescent probes of RNA, and we could say if this expression in the red is more on the green or they are similar based on where you get your RNA. It's the same here. We could, in principle, conjugate the antibodies with molecules that it allows us to detect the antibodies. And therefore, if this antibody is detecting this protein and I'm able to detect that antibody, indirectly, I will detect that protein. So that's how it works. And there are many ways that we can conjugate an antibody based on what is the experiment or what is the application that I would like to do. The first way I could label an antibody, and just be careful that probably the conjugation site is not exactly there. It's just placed there in the scheme, but it doesn't necessarily have to be there. So the first way to conjugate antibodies is to attach a fluorophore. And fluorophores are, in principle, molecules. If you hit with a light, they will emit light back at a different wavelength. So this is a good way to use microscopy, antibodies in microscopy, where you can, in principle, if this antibody detects that protein and you hit with a laser, this antibody will emit light. And in this situation, it's FITC, which is green, then I can detect this antibody in a green color. And therefore, whatever this protein is, whatever this antibody bind to that protein, then I will see a green signal. Okay. You could also use another enzyme that is attached to that antibody, and this is called enzyme conjugated antibody. And the most used in the labs is the HRP, which is the horseradish peroxidase. So this is an enzyme. If you provide with the substrate, it will create a color, and then you can measure the amount of color you can see in that reaction. The more the color that is converted, because of the substrate is converted from colorless into blue probably, then the more of that blue is generated is an indication that this antibody is binding to that protein. And the third one would be the biotin streptavidin. And this is an antibody that we use most of the time if we want to pull down proteins or we immunoprecipitate certain proteins. So say that this antibody is bound to that protein here, and you want to pull it down and see what other proteins probably bind to this, then the biotin streptavidin is the most used. There are others that has different applications. We are not going to go into that one. So the first application that we will describe, and in the next lecture we'll describe many of these applications, is the immunoassay. So in the immunoassays, you have an antibody that is specific to a protein. So this protein is our antigen here, or protein of interest. And that primary antibody is binding to a specific epitope. Now, given the fact that I have different conjugated types that I can use, don't forget that an antibody can still detect another antibody. And this is very, very important to remember, because my primary antibody here is binding to the protein of interest, okay? But my secondary antibody is conjugated, so I can tell every time this secondary antibody binds to something, I can tell how much of that antibody is. So in most of the experiments or the applications I will be showing you in immunoassays, we will be using a primary antibody that is detected by a secondary antibody. And that secondary antibody is conjugated. In this situation, it's like probably a good example of the HRP, because when you provide the substrate, there's an enzyme linked here that changes the substrate. It was undetectable, and now you can detect the product. Now, the more this antibody will bind to the primary antibody, and in other words, binding to that protein, the more the substrate will be converted into the end product, and therefore you will have more of those converted color components. Now, so this is the first application, and probably you have heard about it. It's the ELISA, which is enzyme-linked immunosorbent assay in a plate. It comes in different flavors. There is a YouTube video here for you to check how ELISA is made. But the idea is, in the wells, that these 96 wells, we have this antibody is attached to the bottom of the well. Normally, if you want to do ELISA, let's say to a cytokine like interleukin-1 or TNF-alpha, whatever cytokine that is really available in the blood or the serum or the fluids, or specific to a pathogen or a bacteria, these plates already have a specific antibody that is detecting that antigen, whatever that antigen is. Okay, it's represented here in blue. So this antibody detects that antigen. Obviously, there is another antibody that detects the other epitope of that same antigen. So this is my detection or a primary antibody. And then the primary antibody is detected by the conjugated or secondary antibody. And that secondary antibody has the HRP, the horseradish peroxidase. When you give the substrate, it's gonna light blue. So the more the blue color you have in one condition, say infected versus non-infected, if this is a viral protein, then the more the viral protein is available, the more blue it will be generated. So this is one of the simple explanations of the ELISA. Of course, there are more than one type of ELISA. So this is an image of ELISA plate. There are multiple types of the ELISA. Probably your first antibody is already conjugated toward the antigen. It could be that the primary antibody detects the antigen and your primary antibody is detected by a secondary antibody. This is called indirect ELISA. It could be the one that I just showed you. Two different antibodies detect the same antigen. One is already attached to the plate and one is added after the antigen is captured by the first green antibody here. And then you have the detection antibody, this with conjugation, and that's called sandwich ELISA because the antigen is sandwiched between two different antibodies. And finally, you have the competitive ELISA where you have two different antibodies that detect two different antigens. This is a little bit more complicated for you guys. I'm not gonna go for it. But if we are using HRP, and here's an image of a plate of ELISA, you can see that some of the wells, they have less blue compared to others. So by reading the colorimetric assay, we could tell that this well here, it has more viral protein compared to this one, or in other words, this person is infected versus this one is not infected, or there is bacterial infection here or no bacterial infection here, depends on what is the outcome. So this would be probably everything we will discuss today. Really good, we just stopped at 1 hour. I hope you will have time to go and watch the video here. Well, we still have a few minutes, so probably I'll watch it with you, and then if you have questions, we can discuss.
ELISA is
Okay.
short for enzyme-linked immunosorbent assay. With this plate-based technique, we can detect and quantify soluble substances such as proteins. During an ELISA, the antigen, which is the target molecule, is immobilized and specifically detected by antibodies, which are enzyme-coupled. When providing a colorless substrate, the enzyme produces a measurable, colorful product. An antibody is attached to the bottom of the well. This antibody is specifically designed for binding a specific protein of interest. Another soluble antibody is provided. The antibody is also specifically covering protein X. This antibody is conjugated to an enzyme. When we now add a substrate, the enzyme can convert it to a colorful product, which can be measured later. There are many variants of an ELISA. In the direct ELISA, the antigen is immobilized on the bottom of the plate, and the enzyme-linked antibody directly binds to the antigen. In the indirect ELISA, the primary antibody binds our antigen, and then a secondary antibody binds the primary antibody. The secondary antibody is enzyme-linked. The most common type is the sandwich ELISA. A primary antibody is immobilized and captures the antigen of a solution, and another primary antibody specifically binds the same antigen. But all three ELISAs have one thing in common. The last antibody binding is always conjugated to the enzyme which confirms binding. Here, we provide an example of an ELISA. For this sandwich ELISA, we have two wells, both coated with primary antibody specific for protein X. On the left, we provide a sample containing our protein. On the right, we have the control without the target protein. Our antigen in orange will be immobilized. A washing step will discard everything else. Now we provide the enzyme-linked antibody. It will bind our antigen in the left well and remains bound after washing, whereas the antibody is discarded in the control. In the last step, a colorless substrate is given. Since the enzyme, in most cases horseradish peroxidase, is exclusively present in the left well, it produces a measurable, colorful product here. This can be investigated and also quantified using spectrophotometry. The control remains colorless since substrate is not converted here. If you want to have a closer look into the direct or the indirect ELISA.
Okay. So I think this is everything for today. I'm gonna stop recording.